start with today's episode of Cell. Now, what are the learning objectives of today's lesson? We are going to learn about what is a cell, the discovery of the cell, the types of microscope, the cell theory. So, we are going to discuss what is a cell, the discovery of the cell, types of microscope, the cell theory in brief we are going to discuss. So, we are going to discuss about the cell theory in brief, and then we are going to discuss about the prokaryotic and the eukaryotic cell. I'll discuss about the differences of the prokaryotic and the eukaryotic cells. Now we will divide the cells according to the cell shape and according to the size and number. So we will deal with few examples where we are going to discuss about the cells which have been differentiated according to their shape and according to the size and number. Now let us move on to the next part on the next slide where we are going to discuss about what is a cell. Now here in this slide we can see a typical cell. So what is a cell? We all know that it is a structural and functional unit of life, or it is a structural and functional unit of all living beings. Now here if you follow my red pen, you can see that this is a cell. So this cell is having its organelles and it is a microscopic thing. We all know that we can see cell only under a microscope. So it is not visible with our naked eyes. So any function which is performed by an organism is the outcome of the activities of a cell. It is the smallest part of the body, and a cell is obviously it is capable of its independent existence. All living beings they start their life by a single cell, and the old and the worn out cells are continuously being replaced by the new cell. When the living beings they are starting their life, they are having a single cell, and the old and worn out cells, what are they? They are being replaced by the new cells. Now it consists of an organized mass of protoplasm, and it is surrounded by a plasma membrane. So this is the outer membrane. It is protecting the cell. Now in animal cell the plasma membrane is the only limiting membrane, whereas in a plant cell we can say that outside the cell membrane there is a cell wall. So that we will study in detail. This is in animal cell we are only having cell membrane, but in plant cell addition to this we are having cell wall. Okay, it is clear. So cell membrane is present in animal cell, only cell membrane is present. But in plant cell, with addition to this, we are having the cell wall. So this is in plant cell. Okay, is it clear? Now these are all the organelles which are present inside the cell. Now in the next part of this video, I'll be discussing about each and every part. What is this? What is this? This and all. These are all cell organelles which are present inside the cell. So I'll be discussing about each and every cell organelles with its functions and with its diagram in details in the next part. Now let us move on to the next part of this lesson. So how was the cell discovered, or rather what is the discovery of the cell? Now first, simple microscope invention led the discovery of a cell by Anton von Leeuwenhoek. Okay, it was in 1632 and in between 1723 when he discovered that through a single biconvex lens, and it was known as a simple microscope. Now then only he discussed about the discovery of the first cell. And then came Robert Hooke, who developed a compound microscope. Okay, next scientist to discover the compound microscope with two lenses. So obviously this compound microscope, as it is having two lenses, it is having greater magnification. As it is having two lenses, it is having greater magnification. Okay, is it clear? So it is having two lenses, so it is having a greater magnification. So more the lens, more the magnification. Now next, this Robert Hooke, when he discovered the compound microscope, he discovered that uh, cells were there. So it was almost at the end of 1665 when cells were discovered. Now this cell he studied, how he studied this? He studied it as thin slice of cork under this microscope. So how he discovered cell? He discovered this under a thin slice of cork on a large number of compartments which are joined together. He observed the cork slice and he said that large compartments are joined together, and the structure was how? It was like a honeycomb structure. It was honeycomb-like structure. It was a honeycomb-like structure. And we can say that he named these apartments, these honeycomb-like apartments or structures, as cell. Now next we are going to discuss about the microscope. That is the simple microscope and the compound microscope, the light microscope, the electron microscope, whatever, we are going to discuss about that. Let us move on to the next slide. Now here we are going to discuss about the light microscope. Now the light microscope, what is it? The ordinary light microscope, it is a compound microscope which is mainly used in the laboratories, and it is extensively used in laboratories. It's fairly improved in design by Hooke's microscope, and it consists of mainly two lenses, one eyepiece and the objective lens, which are combined to produce a greater magnification. So two lenses, eyepiece, objectives. Now this is mainly used in laboratories. Now let us now see which one is the eyepiece of this microscope. So this one through which we see. Now let us label this. This is the eyepiece. Now this is the coarse adjustment screw. When we are placing the object on this, so what is this? This is the stage. This is the stage of the microscope on which on a slide we are placing the sample. Now this is the coarse adjustment screw. Okay, is it clear to everyone? If you are not getting any of my points or doubts, please do write in my comment box so that I can clarify that episode, that part or that particular slide in the next video. Now this is the stage where it is kept. Slide is kept. These are two clips which are used to fix the slide. This is the coarse adjustment screw. Here we can see the fine adjustment screw here. But in many microscopes, the fine adjustment screw is here only. When you are working in lab, you will see that there are many microscopes where the fine adjustment screw is here and the coarse adjustment screw is here. Now here we can see the objective lenses are here. These are the objective lenses. These two are the objective lenses. These two are the objective lenses. Okay. So this is the eyepiece through which we are seeing. This is the objective lens. This is the fine adjustment screw, this is the coarse adjustment screw, this is clip, and this is stage. So this is what this is actually, the reflecting mirror. So this is the reflecting mirror. So this is the reflecting mirror. So on the base which it is standing is the stand. It is the stand of the microscope. So this is how you are going to label. If they give you that draw and label a light microscope, so you are going to label it in this way. So eyepiece, objective lens, stage, reflective mirror. This is the stand, fine adjustment screw, coarse adjustment screw, and clips. These are the main parts of this light microscope. Now it is clear till now. Let us move on to the next part, that is the electron microscope. Now electron microscope is not extensively used in labs. Now as you are in 9th standard, you just keep in mind that electron microscope, what does it do? It gives us this times of more magnification. So it is giving us this times more magnification and resolution and much greater than light microscope. Now how it is working? A beam of electrons is passed through this section of the specimen. Now suppose a section of the specimen is kept on the electron microscope. Now it is a beam of electrons will be passing through that. Sorry, there is a mistake here. Beam of electrons will be passing through the section of the specimen and resulting in the photograph, which is called as electron micrograph. So the resulting result is the electron micrograph. So it is a photographic plate for permanent reading. So electron microscope can resolve points up to 1 nanometer apart. So it is very, very, very fine magnification. It is resolving up to points 1 nanometer apart, and in this microscope A beam of electrons is passed through the section of specimen material to produce the image. Okay, so this is all about electron microscope. When you are working in labs with such higher resolution microscopes, then you will be able to see this electron microscope. Otherwise, we are not able to work under this. We generally work under this light microscope, which is easily extensively used in laboratories, which I have shown in the previous slides. Now let us move on to the next part. Now here we are going to discuss about the cell theory. So what is the cell theory? Cell theory is cell is the structural and functional unit of living things. We have already told that. We all know that it is a structural and functional unit of life. Now all living organisms are composed of cells. Now without cells, it is non-living. So without cell, it is not a living thing. It is a non-living. So without cell, it is a non-living thing. It is not a living thing. So it should contain cell. All new cells arise as a result of division of the pre-existing cells. So when there are pre-existing cells only, then by the division or the cell division of that cell only, we get the pre-existing cells. From the pre-existing cells, we are getting the new cells. Now all cells are basically alike in chemical composition. They are having the same type of chemical composition, and they are having the same type of metabolic process. The function of an organism as a whole is the outcome of the combined activities and interactions of the constituent cell. What is it? The function of an organism, how the organism is functioning as a whole, is the outcome of the combined activities and interaction of the constituent cell. So what does it mean actually? How the organism is working? It means that what are the activities or the outcomes of the each cell or the combined interactions of each and every cell, how they are working all together. So this is how we can say that organism or the function of an organism can be decided, that how the constituent cells are interacting, how their combined activity is influencing each and every part of an organism. So this is the basic of cell theory. Now these five are very important point. I think you should learn them properly so that when you are having a question on cell theory, you should write these 5 points as the main points. Now let us move on to the types of cells. Now these are the two types of cell, the prokaryotic and the eukaryotic. Now what is a prokaryotic cell? The prokaryotic cell is a primitive kind of cell. Now it is not having a nucleus, it is having the nuclear material actually in the cells is not enclosed by a nuclear membrane. It is not having a proper nucleus. So you can see that this is a prokaryotic cell. This is an example of prokaryotic cell. So it is a prokaryotic cell, and here we can see the cell organelles. The nucleus is not uh, here. It is not enclosed by a nuclear membrane. There is no such nucleus enclosed by a nuclear membrane. The genetic material equivalent actually to a single molecule of DNA. You can see here the genetic material. You can see actually these cells actually lack several organelles. As in the eukaryotic one, there are no such cell organelles here. They are lacking the cell organelles. Here we can see all the nucleus is prominent. You can see the nucleus prominent. You can see the cell organelles, Golgi aperatures. You can see endoplasmic reticulum. We can see mitochondria. We can see this DNA strands over here. So all the cell organelles which are present in the eukaryotic cell are absent in the prokaryotic cell. So the eukaryotic cell is much more advanced than the prokaryotic cell. The prokaryotic cell, like the bacteria or the blue-green algae, are the examples of prokaryotic cells. So what are the main examples? The bacteria and the blue-green algae. So bacteria and blue-green algae are the examples of the prokaryotic cells. Whereas when we are moving on to eukaryotic cells, we have seen that they are properly having defined nuclear membrane. We are having defined nuclear membrane. Now the genetic material is made of two or more DNA materials. These cells, they have well organized nucleus. They are having mitochondria. They are having the endoplasmic reticulum, lysosome. They are having everything. And they are having the chloroplast, Obviously in plant cells, and in eukaryotic cells they occur also in plants, animals, fungi, protozoa and all. So there are several differences between the prokaryotic and the eukaryotic cell. So what are those differences? Now here in this cell we can say that the nuclear region is very much well defined and it is surrounded by a nuclear membrane. So it is surrounded by a nuclear membrane and it is very much well defined, whereas when we come to this one, there is absence of nuclear membrane and the nuclear region is underdefined. The main thing when we come to the next point is the chromosome. The single molecule of circular DNA is present in the prokaryotic cell, whereas more than one chromosome is present here. More than one chromosome is present here, whereas in the eukaryotic cell, only a single molecule of DNA is present. Now, the nucleolus which is present in the eukaryotic cell is never present in the prokaryotic cell. It's not present here. And the membrane bound organelles, here we can see many membrane bound organelles in this cell, whereas these membrane bound organelles are absent in this prokaryotic cell. Now, is it clear now to everyone that prokaryotic, it is a primitive kind of cell and it is having many many things which is absent in this cell is present in this eukaryotic cell. So prominent is the nucleus, the membrane bound organelles, the chromosomes which is more than one, and the nucleolus which is very much prominent, and the cellular, the main thing we can say that the cell organelles which are present in the eukaryotic is not present in the prokaryotic cell. Now let us move on to the last two slides where we have divided the cell into two based on the cell shape, size and number. Now depending on the cell shape, we can say that there are spindle shaped cells, there are long elongated nerve cells, spindle shaped muscle cells, elongated, their oval or red blood corpuscles are there, cuboidal germ cells are there, branched, osteocytes and chromatophores are there. Okay, now let us move on to the next one. Here we have seen the images of different cells, which is depending on the cell shape. Now, depending on the cell shape, we can say that we have seen amoeba. So this is amoeba which is having irregular shape. So this shape is irregular. Then we are coming to this cell. This is quite long. This is a sperm cell which is actually elongated with a tail, and human sperm cell, it can swim very fast. So it is a sperm cell. Now next comes the oval or the biconcave, the blood cell. Okay, it is the oval or the biconcave shaped RBC which is, can easily pass through blood capillaries and transport oxygen. And here last is the elongated nerve cell. This is the elongated nerve cell. So this is the nerve cell which is elongated in nature. So depending on the different types of cell shape, I have designed this slide. I think you'll like it. So this amoeba is irregular shape. This is elongated sperm cell. This is oval shaped. This is also elongated nerve cell. So these are a few examples which I could find it out and show it in this video. Now next let us see what is there. Now depending on the cell size, we can see that the smallest one is the mycoplasma. The nerve cells are 90 to 100 centimeter, and the bird's egg cell, that is egg of an ostrich, is a very big cell, that is 170 into 135 millimeter in diameter it is. Now we can see that human female ovum, it's also a cell. It is a human female ovum, it is also a cell. It is actually 0.1 millimeter in diameter. It is 1 millimeter in diameter, and this is depending on the cell size, we have divided the cells, and depending on the cell shape, depending on the cell structure, we have all discussed about the cell. Now next video I'll be discussing about the structure of a typical cell. So for today, it's this much for today. And please do write your comments if you are not getting any point, any part which is not clear to you. Please do write in my comment box so that I can clear it up in the next video. So thank you.